many varieties of what's known as trace evidence, hairs have a long history of being used in the attempts to solve crimes. In fact, hair is still one of the most commonly analyzed forms of trace evidence. One of the great values of hair in forensic science is that it's so stable when compared to other body tissues. Hair might be found or analyzed years after a death or a crime, and the recent reopening of cold cases in the United States and elsewhere has shown the stability of hair as trace evidence over long periods of time. But let's begin by stating right up front what hair analysis can do and what it cannot. Standard hair analysis cannot conclusively match two hairs. So then, what good is hair in forensics, and why is it still so useful? Many people think that since DNA technology has come along, it's eliminated the need for other forms of testing, like forensic hair analysis. But consider a case where, say, a woman was at a big party and claims to have been raped on her way home by a guy who was also at the party. The victim shows up at the emergency room, and the sexual assault nurse examiner takes the woman's clothing for submission to the crime lab. The victim says that the perpetrator wore a condom and there are no obvious semen stains anywhere. So back at the lab, the trace evidence examiners find several head hairs on the victim's clothing, and in her panties are a couple of pubic hairs. None of these hairs seems to be a visual match to the victim's own hair in terms of length or color. So the police then round up the suspect, but he says he has an alibi. He claims he left the party with somebody else and spent the whole night with her. He says he never touched the lady who said she was raped. Now, do you have any idea how long it takes to get DNA samples back from the lab? On TV, they get them back after the commercial break, right? But in reality, it usually takes months or even longer. Besides, it's not always possible to get DNA out of a hair sample. So what can investigators do? If the suspect is willing, or sometimes by a court order even if he's not, the crime lab can get a sample of his head and pubic hair for comparison. The suspect's hairs are the known hairs, which my trace examiner friend refers to as the K sample. And the hairs on the victim's clothing are the unknown hairs, called the Q sample, Q being for questioned. Well, if microscopic comparison reveals clear differences between the K and Q hairs right then and there, that guy from the party is no longer a suspect. Without going to all the taxpayers' expense of DNA testing, and without months passing while an innocent man stands accused of something terrible, the crime lab has conveniently eliminated him from suspicion just by looking at some hairs under a microscope. That's the power of exclusionary evidence. But what if the hairs are similar? Does that prove the man is guilty? No, but it'll certainly allow investigators to continue to focus on him as a suspect and gather additional evidence to either show his guilt or support his innocence. Microscopic hair analysis only serves as class evidence. That is, remember, evidence associated with a group rather than an individual. But if the hairs are similar, DNA testing will probably be one of the next steps taken. Now that we understand something about the value of hair as forensic evidence, let's take a closer look at its structure. Now that we've learned something about forensic hair analysis, let's summarize the specific steps involved in a hair comparison. First, a questioned hair has to be identified among all the evidence it is seen, and that can be difficult in itself. Then that unknown hair has to be classified as to its type, like is it head hair, pubic hair, and then be assessed to see what tests it's likely suitable for. For example, does it have a follicle that could yield nuclear DNA, or does it not? Next, a representative group of known standards of the same type of hair, head or pubic, have to be taken from a suspect or somebody that needs to be excluded. If there are similar features, they should be noted even before a microscopic exam. If there are not similar features, then the comparison's done and the report gets issued. But if the questioned and known hairs warrant a complete comparison, then they do have to be examined microscopically. To do this, analysts typically embed each hair to be compared on a microscope slide in what's called a mounting medium. Now, if you just put a hair on a microscope slide and looked at it under the scope, or examined it using a water mount, where you just put water on the slide, you really won't see the details of the cortex and the medulla. In order to allow light to penetrate the hair so that the details in the cortex and medulla can be seen, investigators use a mounting medium with a refractive index similar to that of hair itself. Remember, we talked about refractive index when we talked about fibers in glass. So then, using side-by-side comparison microscopes, detailed observations can be made using all the features of the cortex and its pigment distribution, as well as looking at the medulla and its form. If there's a positive association between the two hairs being compared, my trace examiner friend says she always gets a second opinion from another qualified hair examiner. This is because of the subjectivity involved in drawing conclusions by comparing two hairs. In fact, there have been an unfortunate number of forensic cases, including relatively recently, where investigators really stretch the boundaries of what can and cannot be known from hair analysis. Most often, these cases are related to a single hair from a crime scene being linked to a suspect, allegedly at the exclusion of nearly all other individuals. In other words, more was made out of the evidence than was scientifically possible, often accompanied by unsubstantiated statistics. Take, for example, the case of forensic analyst Joyce Gilchrist, who was employed by the Oklahoma City Police Department for 21 years and participated in thousands of forensic investigations. Evidence she analyzed, including numerous hair comparisons, contributed to over 23 death sentences. Tragically, 11 of those people had already been executed by the time her conclusions and the methods she was using were seriously called into question. Through DNA testing, they've now exonerated some of Gilchrist's victims, and case reviews has shown that she either lost or destroyed evidence, and she made specific conclusions about hair comparisons that were completely unwarranted. So, what kind of valid conclusions can be made from hair comparisons? My trace evidence friend put it this way. She said, if she could take a couple of questioned hairs, remember those are her Q hairs, and mix them up with a sample of known hairs from someone, those are her K hairs, and then shake them up, dump them out, and not be able to tell them apart even after using all of her cool tools, then the most she can say is the Q hairs are consistent with originating from the known person. But it also has to be understood, especially in court, that those same questioned hairs could also have come from any other person whose hair exhibits all those same characteristics. An investigator really then has only three options with regard to reporting on the comparison between any two hairs. And the same goes for lots of other types of evidence comparisons. First, if there are no unexplainable differences and lots of commonalities, a trace analyst may conclude that the two hairs could have come from the same person. Secondly, if there are clear differences that go beyond the typical range of variations seen in a single person, then an analyst may conclude that those two hairs could not have come from the same individual. And third, if they see both similarities and differences between two hairs being compared, then no conclusion can be made about a potential association between the hair in question and hairs taken from a known person. This is because hairs exhibit features that are shared among a group of people, which again means we're talking about class evidence. And the size of these groups that share hair characteristics isn't even close to being known. Investigators have attempted to classify hair in much the same way fingerprints are categorized. But human hair really can't be fit into any reasonable number of classes to allow for the same discrete categories like they have with fingerprints and, as we'll see, with genetic markers in DNA. So there's just no way to establish probabilities among individuals and say, for example, that the chances of two similar hairs coming from two different people are 500 to 1 or something like that. The other problem is that when people change their hair, or as it changes naturally when they age, the hair then changes class groups when it changes its features. This is different then from fingerprints or genetic markers, which don't change as we age. So, because of the inherent difficulties and the subjectivity involved in hair comparisons, only a highly skilled and very experienced trace evidence examiner can make a quality assessment of the similarities among human hairs. This is not only because there can be strong similarities between the hairs of two different individuals, but also because there can be striking differences among multiple hairs taken from the scalp of just one person. In fact, you might remember that one of the questions raised at the famous O.J. Simpson trial was how representative a single hair is with regard to the hair on a person's entire head. The answer to that question seems to depend a lot on the person. To come up with a good hair standard for a single person, many known hairs are needed. That's because even on the same person's scalp, any number of hairs might vary in diameter, length, and color. Protocols differ, but I've seen it suggested that an investigator should collect at least 50 full-length intact hairs, that means hairs with the root, from the scalp of a single subject to serve as that person's known reference sample. And because of regional differences, the hair should come from a variety of locations on that person's head. In rape cases, it's suggested that at least 20 intact pubic hairs be collected from a suspect before attempting a comparison with an unknown pubic hair from a crime scene. But more is better, and the FBI recommends 100 scalp hairs for a known standard. And the RCMP, that's the Royal Canadian Mounted Police, suggests pulling 150 sample hairs. Man, that's gotta hurt. So, where does that leave us? Similarities between hairs can be noted, and dissimilarities between hairs can definitely provide exclusions. But there is currently nothing short of nuclear DNA technology to positively link a hair to a specific person. We have a DNA lecture coming up shortly, but let me highlight some of the fundamentals now. There are two basic types of DNA found in human cells, nuclear and mitochondrial DNA, and both are relevant to hair analysis. Nuclear DNA is inherited from both parents, and it's unique to each one of us, except in the case of identical twins. Analysts can get nuclear DNA from follicle cells surrounding the hair root, or sometimes even from the actively growing anagen root itself. But remember, follicle tissue is only found on hairs that have been forcibly removed. Hairs that are on their way to being naturally shed are in the catagen and telogen stage, and they don't have actively growing roots. On the other hand, mitochondrial DNA, which is inherited only from a person's mother, can be obtained and analyzed from a hair shaft. The problem with mitochondrial DNA, though, as we'll see, is that it is not as discriminatory as nuclear DNA, since it's shared among all people in a maternal bloodline. It certainly does add additional data to a case, but it's not considered positive identification. In 2002, the FBI published a study that used mitochondrial DNA to try to assess the validity of microscopic hair analyses that they conducted between the years 1996 and 2000. Their self-study looked at 80 so-called positive microscopic associations that qualified FBI hair analysts had made and compared them to mitochondrial DNA analyses on those same hairs. The results showed that 11.25 of those microscopic associations made by the FBI were, in fact, not true associations. Now, that doesn't mean the analysts were wrong. The hairs themselves did show positive microscopic association. It just means those associations were not valid when compared to DNA testing, and reminds us that hair is class evidence. That study is an example of good science and critical self-analysis. It doesn't negate the usefulness of hair analysis, since nearly 90% of those FBI associations did hold up. But it certainly suggests that all evidence comparisons, not just hair analyses, but all of the types we've covered and those yet to come, should only be undertaken by the most experienced and ethical of professionals, and always backed up with additional